Hi students, so today is 20th January of 2021 and we are this video we are going to talk about Form 1 Science Chapter 1 about microorganism. So we are going to the category of bacteria. So we already learned in the previous video got virus, got bacteria, got fungi, got alga and fungus. So now we want to talk about bacteria. Basically, all is almost the same because it's microorganism, very small organism. Only different is some have chloroplast, some can do photosynthesis, and like for virus is very small, even smaller than the bacteria because virus is the smallest microorganism. So a slight different, but it's like still the under microorganism. So bacteria, what is bacteria? Bacteria can be seen under the light microscope around 0.2 to 10 UM. That's a nano, right? U is nanometer, right? Means times 10 power of negative 9, if I'm not mistaken. Negative 9 or negative 12? I think this one is negative 9 times 10 power of negative 9. Means 1 meter times 10 power of negative 9. You know lah, if you know mathematics. So these are the example. I think the most famous one is this one, coccus. And then Bacillus. Usually exam they won't ask the top part the name, but if your school teacher asks means you do something back to your teacher already. The teacher want you to write this. Okay, the name of the species, Streptococcus sp dot. Anyway, exam won't ask so don't bother to remember that. Remember this one. As I say, don't make the teacher angry. Your school teacher angry, they give all these weird question. Basic structure of bacteria. Okay, so again, we have the phloem, cytoplasm, cell wall, cell membrane, and glycogen. Okay, so we can see from here, it has a cell wall which gives the shape. Because usually it's the plant cell, right? They have cell wall, but I will consider this is more towards animal cell because it don't have chloroplast. I don't see any chloroplast at here, but it got glycogen, which also in human. If I'm not mistaken, glycogen is like the fats, the oil, the glycogen. The cell wall is not made out of cellulose, but it's made out of amino acid. So it's called protein or a type of protein. So next time after the science class, you don't say fish, I want fish. You don't say I want protein. You tell your mom, wow, today mom you cook amino acid lah. Wow. Or you cook polysaccharide so nice. Then you can teach your mommy science. And the structure known as pili, and they have the shape called flagellum. I don't know how to read this. Flagellum. This one is like the leg to help the movement of the cell. So this is the leg. But you don't write the leg in the exam, okay? You say, what is the function of flagellum? Flagellum. So, what is your answer? I mean, the question, I say again. What is a flagellum? Very good. You got the answer correct, okay? It is a structure shaped like a tail to help in their movement so they can move. Very good. The student is very smart. Nutrition. So nutrition, bacteria which have chlorophyll. Of course, you have chlorophyll. You produce your own food. Can you imagine that if I got chlorophyll, I don't need to eat it. I just need to stand under the sun, then I become full already. But I become green color like the heart because I got the chloroplast. Some bacteria are parasite, useless people. Let's go like parasite. Always suck people nutrient on it, suck people work power. Some are saprophyte. Still remember last time we got learned a few relationship. Parasitism, you steal things from other people. Saprotism means you take food from that organism. Mutualism, because one benefit and one don't have benefit. Some more got Any more relationship? I think got one more. What's the name already? I forget already. Okay, let's continue. Habitat. Okay, so we already learned from previous video. Got binary fission. Got conjugation bisexually. Asexually is by binary fission. So I just draw this one. And sexual reproduction is by conjugation. But of course, human is called internal fertilization, right? So that's why it's internal. You know why it's called internal, right? Okay. Third conjugation, then you can see from here again, conjugation, formation of conjugation tube, contact between two cells, kiss, transfer of genetic material, and you got the identical daughter cell. Then got the daughter, got baby already, got daughter cell. So we can see this one is endospore. We can see from the word spore, right? Means the wind blows. You remember? Got budding, conjugation, binary fission, and spore formation. Do you remember spore formation? Like the wind blow and then the plant spread or the animal spread, like the virus MCO, right? It travel through water droplet lah, but also through air, right? Correct or not? That's why we need to wear the mask. So spore form in the bacterial cell which have strong protective coat. You know coat? This is like this is also a coat, right? A shirt coat. Okay, so it's surrounding it to have a favorable growth. And of the endospore become the favorable for growth. This allows germination of new bacteria. So you can see there's a protect this layer. Protect the spore so that it won't spoil. Until they got the opportunity. This protective coat will rupture when the surrounding or endoscleral is favorable. What do you mean by rupture? Rupture means like gone lah. So correct place, this thing will disappear. And then this one can become the new microorganism. So this one gone. On how to make this gone? With when the surrounding is favorable for growth. Maybe like got water, got good temperature, then it will start to grow. Okay, no question. I think this one is the last one, is it? Virus, like MCO, coronavirus. So small, so small, we call it smallest, 0.5 UM. Only can be seen not by light microscope, because we got a few microscope, right? We got like your laboratory microscope, we got your light microscope in the in the laboratory, we got electron microscope. So like in the terms like electron, like it's very small until like can see electron. So this is the electron microscope. I think this thing is very expensive, so you cannot find this one in your school. Okay, so let's look at this one, influenza virus. Do you remember like H1N1, right? Influenza H1N1. After that, we have something like this. We have something like this, or we have something like this. So what happened is the virus will infect another cell, like this one. You can see that the virus actually go into the cell. So this, you can see this is bacteria, you know. So usually it's like the virus can even attack the bacteria. So it will go in, change the genetic, change it, and then come out four baby break and continue to split. This is how virus duplicate itself, and it's so dangerous because you can see that it will attack your cell, it will modify your genetic. You can see here, virus injected a genetic material, the the DNA into the bacterial cell. So it actually changed your DNA to a certain extent so that it start to reproduce. Like cancer cell like that, correct or not? Cancer cell. Cancer cell also is a bad cell, but somehow it continue to duplicate and your body unable to stop it. Or maybe your body start to don't know that it is a cancer cell. So it start to develop until the person gets sick and die. Let's continue on activity. Okay. The presence of microorganisms. So where is microorganism? Microorganism is at everywhere. Okay, that's why we need to always wash our hand and try your best not to use your hand to touch your face. That's why you know, right? Coronavirus, don't use your hand to touch the face because usually if your hand dirty, when you touch your eye, the bacteria will go into your body or your mouth or your nose. So I don't touch your face. Even when I go to toilet, I tend to use this to push because I don't want to dirty my hand. So now MCO, right? So I go to toilet, I push, or you use this finger to open the door. Rather than you hold like this, the chances is higher for you to touch your face. So I just open the door like that, or when I push the door, I use my shoulder to push the door. Because I go to McDonald's toilet, I'm very scared to hold the door. I scared like a lot of people is never know the last person didn't wash hand, right? So I just push the door on it. Okay. So this is an experiment to see the bacteria growth, nutrient growth. Okay. So what is the hypothesis of this experiment? Okay, this experiment is very long. Okay, the shorter way of saying this is, let's put this. Here it say that, and then I just highlight. Bacteria growth, the relationship of bacteria growth and the cleanliness. That actually two things. I mean, like, you want to make this hypothesis simple, it's supposed to be like, the cleaner the surface, the less bacteria growth. So it should be simple, okay? Or you can make it longer. So, you want to do the manipulated variable is the cleanliness, responding will be the growth of bacteria. Or for this one, you can also type growth of bacteria, right or not? Growth of bacteria. So it's the same thing, right? And surrounding temperature is the constant. I don't want to go into this, I think you all know already, right? So even though without doing the experiment, we know that more clean means less bacteria, more dirty is more what? So for that, for example, this one, petri dish, and how you want to observe it? Is it by ruler? Observe it in the temperature and see. Number of bacteria colonies, streak with unwashed finger, with no streak with any finger, streak with finger that have been washed, streak with finger that have not been washed. So if no touch finger, so even though I didn't do the experiment, but we can predict the result. Can you predict the result? Okay, so we can see from here, no streak with finger. No streak means here is clean, right? Streak with finger that have been washed. So this one is clean a bit. Streak with finger that have been washed with water, not clean. Streak with unwashed finger, very dirty. So this one is the number of bacteria growth. So for the clean one, I put 0 lah. For this one, I put 3 lah. You like lah, you put 4, 5, 6. But this one must be more lah, because this one is more dirty ma. So I put 5 lah. This one I put, don't put like 100 lah. Maybe you put 7. Correct or not? If you don't put 100, then it just doesn't make sense, right? But or you can put 0, 1, 2, 3 also can. or 1, 2, 3, 4 also can. I mean the answer must be like logical, okay? Because here say the number of colony means the more dirty the thing, the more bacteria growth. The cleaner the place, the less bacteria grow. That is why when we go to hospital, we like hospital to be clean, right? Because hospital people get sick already, you still don't want to be clean, then people die of bacteria infection. So you can go to, I like to go to hospital because I think the floor is so clean that you can sit and sleep on the floor. So it's very, very clean. Conclusion, is the hypothesis accepted? So what is the conclusion? So the conclusion is same as hypothesis. Don't worry, because from 1 until from 5, your answer must be yes. Unless you go to university, you do your own experiment, sometimes the hypothesis is no, but it must be something. But for from 1 to from 5 is always yes. So hypothesis is yes, hypothesis accepted. Then you just copy the hypothesis, which is this one. When the cleanliness level of the finger restrict the surface of the sterilized nutrient agar increase, means more clean, correct or not? Sterilize means you like you put hot water to, like the doctor when they want to do operation, they need to sterilize the equipment so that it's hot, it's clean. That before you, like Chinese people, you go and eat, also you put in the cup hot water, right, to sterilize it, to clean it, to kill the bacteria and to clean it. So if the level of cleanliness increase, the bacteria growth of the sterile nutrient will decrease, correct or not? Means the more cleaner you are, the lesser the bacteria. So yes, so this is what we call science, right? Because science is not like, yeah, you say, I say, you think, my father think, but this one is an experiment, really proof that the cleaner the surface, so there'll be less bacteria, less sickness. So we must make sure people to be clean. That's why we ask people to wear masks, to wash hand, wash leg, take bath, and so on. What are the factors that affect the growth of microorganism? Humidity, light, temperature, and pH value. For example, like temperature. If too hot, can survive or not the bacteria? I put the bacteria in the hot sun, so maybe 10 minutes or 1 hour die already because the sun is so hot, especially in Malaysia. And also the light, the temperature and the light. If I take the bacteria put in the toilet, I think the bacteria will survive for very, very long. Maybe can survive for 1 week, 1 month, or 1 year. Humidity means the water in the air. For example, Malaysia is what? Malaysia is hot and humid. India is hot and not humid. Means the air now got water, humid. So I feel more comfortable. Maybe because you in Malaysia we always rain. So we always got hot sun and the air is humid. 很舒服，有水. But when you go to a place that is less humid, you will feel like itchy, or you always feel that you are sweating because the humidity is not there. That is why people you go to supermarket, they buy the humidifier, put the water in the supermarket. So when you turn on the aircon and then you on the machine, got the water coming out, then you can sleep better, and it's better for your breathing also. Okay, pH value depend on the bacteria. So here it says 7 is the most optimum value for the pH value. Too high die, too low die, correct or not? Too low is 0, right? Too high is 14. So 0 is like acid. You put the acid, of course the bacteria die lah, huh? Or the alkaline also, too alkaline also become acid. You put the alkaline. then the bacteria also will die. Okay, so we have humidity, we have light, we have temperature, pH value, and nutrient. So these are the 5 things that microorganisms need. So in the exam, they will ask if 1, 2, 3, 4, 5 effects that affect the growth of microorganisms. You need to name the 5. Humidity, light, temperature, pH value. Okay, so this is the experiment. I think we are going to skip the experiment. And this is another experiment. Effect of humidity. Okay, of course, one you put got and you put don't have. If got humid means more growth, no humid means less growth. Okay, skip this one. We have the effect of light. I think one is in the dark room, one is in the light room. If it's too bright, it depends on. Wait, let me read for a while. Okay, in the bright area near the window. Here say the bright area near the window. So this one you put in a dark cupboard, this one you put in a bright area, and you see how does the number of colonies grow. So we can see a hypothesis. Light retards the growth of bacillus. Means if got more light means no bacteria grow. Correct or not? Like the bacteria you put under the sun and you put in your toilet. Of course the toilet one will grow. Under the sun got light, usually it don't grow. That is why when in your room, maybe you want to open during daytime, you open the window, you open the sunlight to kill the microorganism. Or your shirt, you want to kill the bacteria, or your pillow, you want to kill the bacteria, you go put under the sun. And then after that, you take back your pillow, you smell, very nice. Because all the bacteria die already. The sun already killed the bacteria, correct or not? Not only will kill the bacteria, will kill you also. If you stand there for 1 hour, you see, you can die, right? Stand for 10 hours and see under the sun, hot until you die. So, effect of temperature, this one you know lah, huh? One you can put in the fridge, one you can put in the room temperature, one you put in a hot place. And for this one, we can see that the effect of growth of bacillus, or what is the hypothesis? Growth of bacillus at the highest is the highest at room temperature. So they want room temperature. Like we prepare food, either you want to make it cold, the soup, you put in the fridge. Then the soup you can drink tomorrow. Or because it's too cold, the bacteria cannot grow. Or you put it hot. So like the curry, you don't want it to spoil, you want to cook in the morning, you want to keep at night, you always cook the curry for like a while, wait. After 2-3 hours, you boil again the curry and wait. After 3 hours, 2-3 hours, you boil the curry and then wait. So at night, even morning, you eat the curry will last until that night. Because you already boiled, the temperature is hot. Skip. pH value, I think you can do this by yourself. Okay, let's do some of this exercise. Okay, so that's it for this video and I see you next time. Bye bye.